morning, dear students. In the last class, we have learned about the acrosome reaction. And today's section, I am starting with telling some more detailed aspects, some more additional changes that is happening to the sperm head during the acrosomal reaction. Okay, we know that during this acrosomal reaction, the sperm is binding with the zona pellucida. And what is acrosomal reaction? I am just checking your memory. Acrosome reaction is the process of fusion of outer membrane of acrosome with the sperm membrane. And because of that fusion, the content in the acrosome can discharge outside. And it contains so many enzymes that will help the sperm to traverse the zona pellucida and to reach near the egg plasma membrane. Okay, and as the outer membrane fuses with the sperm's membrane, the inner membrane of the acrosome get exposed to outside. And in the last video, we have seen that the inner membrane contains so many binding proteins. As this inner membrane get exposed to outside, this binding protein also get exposed to outside. In the meanwhile, I am going to explain you some other points also. That's what I am just going to draw roughly a sperm structure. We know that here we can find the acrosome membrane, and behind that present so many actin monomers. So many actin monomers. And also they present the sperm nucleus. And during the time of this acrosome reaction, we know that the acrosome reaction is occurred because of a spike in calcium ion level inside the sperm. And the same calcium ion spike results in the polymerization of this actin monomers and resulted in the formation of actin filament. And that process is called the acrosomal process. And as the inner membrane of acrosome get exposed to outside over this acrosome process, we know that the binding protein that present in the acrosome inner membrane also get exposed and can now remain very close to the egg plasma membrane. And the next step is the fusion of sperm membrane with the egg membrane, right? For that fusion, there requires some proteins interaction. In the case of sperm, there present a protein. Its name is isomo. And in the case of egg plasma membrane, there present another protein. Its name is CD9. This CD9 is an integrin associated protein. And these two proteins interact each other, and as a result of that interaction, the sperm membrane gets fused with the egg membrane. Understand? Okay, at this point, there is chance to happen polyspermy. Polyspermy means a condition in which more than one sperm come and fuse with the egg. So, it is highly unfavorable. We need to prevent that. And we take the case of organisms that are having external fertilization. There are two methods to prevent polyspermy. First is fast block and the second is slow block. Okay, and this fast block give only a temporary protection and the slow blocks give a permanent protection against the polyspermy. And in the case of organism having external fertilization, they are in the risk of encountering with so many sperms, and also those sperm may come from different species. So, it need very urgently to prevent the polyspermy. And so, that fast block is significant in the case of organism having external fertilization. Why? there is a reason, because we know that this low block is a permanent solution for polyspermy. It requires some time to develop. So until that time, the egg need to be prevented or protected against the polyspermy. So that is why the organism that are having external fertilization, that a fast block also present to prevent polyspermy. As we are talking about mammalian fertilization, as it is happening internally, so here the slow block is relevant. This fast block is less significant. Okay, clear? And so I am going to explain here next how a slow block is developing in the egg to protect polyspermy. And while the sperm membrane fuses with the egg plasma membrane, an enzyme named phospholipase C. Phospholipase C is released to the egg cytoplasm. And what this enzyme is doing? This enzyme come and cleave a phospholipid. Its name is, I will write here, phosphatidyl inositol 4,5 bisphosphate. In short, PIP2. So that phospholipase C enzyme come and cleave this phospholipid and results in two products. One is DAG and the other one is IP3. Okay, DAG is diacylglycerol. IP3 is inositol 1,4,5 triphosphate. It is the primary candidate that results or that helps in the development of slow block. How it initiates a slow block? This IP3 moves to the endoplasmic reticulum. Endoplasmic reticulum inside the egg cytoplasm. It moves inside the egg cytoplasm and results in the efflux of calcium store that present in the endoplasmic reticulum to outside. By that way, it results a calcium surge, a calcium spike inside the egg cytoplasm. And as a result of this calcium wave, the cortical granules, these are all cortical granules, it comes and fuses with the egg plasma membrane and empties its contents to this pre-vitelline space. And it contains so many enzymes. And one important enzyme is, I will write the name here, N-acetyl, some problem with the pen, glucosaminidase. N-acetyl glucosaminidase. This enzyme is important. What this enzyme is doing? It removes the carbohydrate moiety of that glycoprotein that present in the zona pellucida that serves as a receptor for the sperm. So by that way, it prevents any further binding of a second sperm. Clear? So this enzyme come and cleave the carbohydrate moiety of ZP3 glycoprotein that present in zona pellucida. By that way, prevent any further sperm to come and bind with the ZP3. And which carbohydrate moiety is cleave of this ZP3? This enzyme cleaves the N-acetyl glucosamine. N-acetyl glucosamine residue of this ZP3 glycoprotein. Okay. And they present some other proteases inside. What they are doing? They clip another glycoprotein. We know its name is P2. Is that P2 that present in the zona pellucida? That also serve as a seed for or yeah, a sperm to come and bind with. So in that way, that glycoprotein also blocked to bind with another sperm. This is the slow block. This is the slow block against polyspermy in the case of organism having internal fertilization. And also one more thing we need to understand that those organisms that are having internal fertilization and a fertilization envelope is not forming. Fertilization envelope is not forming in the case of organisms that are having internal fertilization. But in its slow block, it also very much efficient to prevent polyspermy. That means the effect is same. Even if fertilization envelope is not forming in the case of mammals that are having internal fertilization. Clear? Next, we are going to see the fusion of genetic material. In this picture, we can see that the sperm's membrane is fusing with the egg plasma membrane and releasing its nucleus to the egg cytoplasm. And as a result, inside the egg, now we can see the male pronucleus and the female pronucleus. The male pronucleus is in haploid condition. But the female pronucleus, it didn't finish the meiosis. Instead, it is stalled at the metaphase 2. Okay, we know that during the fusion of sperm's membrane with the egg plasma membrane, that time there develops a calcium surge or a calcium wave, and this activates a kinase. And what this kinase will do? It proteolyzes to protein. To proteolysis two proteins. One is called securin and another one is cyclin. And what is the significance of securin? Securin is a protein that holds the metaphase chromosomes together. And the proteolysis of the cyclin, it helps that female pronucleus to proceed the further steps in cell cycling and to attain the haploid stage. Okay, clear? And along with that, the same calcium ion surge, it inactivates another protein. Its name is MAP kinase. What is the significance of this protein? This MAP kinase actually it inhibits the DNA synthesis. As this protein is inactivated, DNA synthesis takes place in both pronucleus. And as a result, now this pronucleus chromatin is having two sister chromatids. Okay. Along also, as the sperm nucleus enters the egg, along with the sperm nucleus enters the centrosome of the sperm to this egg cytoplasm. And what is centrosome? What is its role here? It produces the aster tubules and it extends and it drags the female pronucleus towards the male pronucleus. As it drags the female pronucleus, it assumes the shape of a drop and it is dragged towards the male pronucleus. Okay, in the case of sea urchins that are having external fertilization, as these two pronucleus meet together, its nuclear membrane dissolves and they fuse together and will form a diploid nucleus. That means the sea urchin is having a diploid condition. It is showing a diploid condition from the zygotic stage onwards. But in the case of mammals, here as after these two pronucleus meet together, here also the nuclear membrane dissolves away. At the same time, this centrosome is splits to form two centrioles and orient conveniently to form the mitotic spindle. Also, along with that, that chromatins that present the chromatins from the male pronuclei and female pronuclei both condenses and it align with the mitotic spindle in the metaphase plate that we can see in this picture. In this way they align, and it undergoes the first mitotic division and results a two-celled stage. So, in the case of mammals, the diploid state is attained or complete diploid state is attained only in the two-celled stage. I will show you a picture. In this picture, again you can see the sperm enters the egg. It is stalled at the metaphase 2. As the tubules are developing, is dragging the female pronucleus towards the male pronucleus. They are meeting together. The nuclear membranes are dissolving away, mitotic spindles are forming, and the chromatin condenses to form chromosomes and align with the mitotic spindle in the metaphase plate. Okay, so more clearly we understood that in mammalian egg cells, the chromatin from the pronuclei they condenses to form chromosomes and merge with the mitotic spindle. Okay, and the diploid nucleus in the case of mammals started to see from the two-celled stage onwards only. But in the case of sea urchins, the diploid condition is present from the zygotic stage onwards. Okay. Along with that male pronucleus that entering the egg, certain other structures are also coming from sperms to the excitoplasm. That includes mitochondria. But the mitochondria that entering the excitoplasm get degraded along with its DNA. That means whatever mitochondria that present in the zygote, all those mitochondria are obtained from the egg, not from the sperm. Clear? Then what else come to the excitoplasm? A little cytoplasm of the sperm, and some proteins and mRNAs are also coming. An example for the protein is phospholipase C. Lipase C. I think that you remember the significance of this protein because it creates the calcium ion wave inside the excitoplasm that will bring about a significant process in that gamete fusion as well as the genetic material fusion. Okay. And also one miRNA, microRNA also entering the excitoplasm from the sperm. It has some regulatory role in cell division of the embryo. Okay. Next, we are going to see the significance of fertilization. What are the significance? The fertilization helps to restore the diploid number of chromosomes again in the individual. That is one significance. This fertilization provide a stimulus for the ovum to complete its maturation because we know that the ovum is previously stalled at the metaphase 2. So it provides a stimulus for the ovum to complete its maturation. Also, the fertilization helps to combine the characters of two parents. By that way, it brings about recombination of genes and introduces variations. Okay. And fertilization helps to determine the sex of the embryo in the humans. And also, it introduces the lost centrioles again in the ovum. And still now it's not clear how the ovum loses the centriole. Anyway, fertilization helps to introduce the lost centrioles again in the ovum. Okay, with this, we finish today's section and class. Thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in a Benz.